Our next speaker is Joseph Wang, Chair and Professor of Nanoengineering at UCSD, and the Director for the Center for Wearable Sensors. And a very interesting fact about Joe is that he's authored over 1,070 papers.
Sign of aging, huh?
No, sign of brilliance.
Let's see. Have some IT support to switch the computer. Who is the IT support? Need to switch. Oh yeah. And Mike, let's see. You can hear me coming from warm San Diego, 50 degrees north, 75 today. Our sensor on the skin. So this is digital health, wearable technology, but what we have on our gadget is vital sign. Testing, yeah. So currently only we can measure steps and ECG and other vital, but what is missing is the chemistry. So we would like to add biochemistry to filling the gap of my pointer here. Anyway, yeah. We want to fill the gap by getting chemical information, not only glucose, but many other biomarkers. So this is what you have now, ECG, motion, temperature, respiration, but we would like to add the biochemical marker ranging from glucose to cortisol to alcohol to lactate, electrolyte, other metabolite, and many more. So this is the opportunity, but this is also the challenge because you depend on enzyme, which depend on the temperature. You have biofouling issue. This is why we don't have a glucose on my iWatch on the Samsung. So this is everybody dreaming to have a glucose on the iWatch, but it's still the challenges of stability and fouling. So these are the challenges. The benefit is that you can get continuous chemical information. But anytime, any location, this is non-invasive. You don't need to pierce the skin or the fingers, and it will change the way wellness, self-care, and health is being monitored. Again, for self-care, wellness, and medical, also security application, elderly remote monitoring, lower health score, and so on. So the challenges are the stability of this sensor on 24/7 under a changing condition. We need to validate compared to the gold standard, the blood testing. We need to do it in a safe manner. If you do it in the mouth or on the eyes, we need to meet the accuracy of standard lab testing. Some of these assays, like immunoassay, need the complex steps like washing and tagging, which is not easy on the body, issue of the mechanical strain on the body, and data security. In addition, if you're using enzyme like for glucose or lactate, glutamate, there is the stability of the enzyme, the sweat pH is changing down to 5. The temperature is changing if you are jogging in Arizona in the summer at 110 degrees. So these enzymes are not meant to survive this extreme condition, changing condition. So we use electrochemical sensor, like this is what Dexcom is using for CGM, continuous glucose monitoring, all the blood glucose meters. So electrochemical offer many advantages of high performance, low power requirement, miniaturization, and ease of use. So these are some examples of the diabetic strip, a multi-billion-dollar industry. This is iStat for emergency care, but these are all in vitro testing, lab testing, this on-body Dexcom for glucose. So this is a Amperometric, measure the current signal of glucose, or potentiometric, potential signal for pH or sodium hydration, etc. So this is from my Nature Biotech review, the evolution of biosensor from the 60s the first Clark glucose sensor over the years. ELISA was introduced in the 70s, glucose strip in the mid-80s, DNA sensing in the late 90s. And then the last decade is the wearable on-body testing that you see a lot of progress. But again, it started with vital sign, and only recently we moved to chemical sensing. So if you visit my lab, you see this mannequin with many, many sensors in the mouth, on the skin, on textile, on socks, on glove, on ring, and we will cover some of these. The goal is wear and forget anytime, any place for chemical markers. So we'll discuss this main platform, textile-based sensor, a microneedle, which are minimally invasive, skin worm tattoos, a mouse guard for saliva monitoring of metabolite and electrolyte. So these are the biofluid you can do non-invasively or minimally invasively, so sweat, saliva, tear, interstitial fluid. Also we monitor the surrounding environment for security, safety, and environmental. But most of the work is for biomedical and wellness application. So this is the type of molecule, biomolecule, electrolyte we can monitor. And glucose is still the top of the list because of the marker. Hydration, sodium, potassium, pH, drug, we do a lot of opioid, fentanyl, L-DOPA for Parkinson, stress marker like cortisol, various disease biomarker. We work on vitamin C, D, various nutrition company, inflammation, cytokine, IL-3, TNF-alpha, neurotransmitter, different other substance of abuse, chemical threat for the DOD, nerve agent, explosive residue, and toxic metal. So we'll start with a printable device for textile and skin. This is the screen printing, which is widely used in this glucose strip. But these glucose strips are very rigid, brittle. They are not flexible that would like to do on the body. So the way we print, let's say on a textile, this is a Navy project for different injuries, traumatic brain injuries, soft tissue injuries. So these are 8 sensors for different injury. We print on a Gore-Tex for military. So we have the design of the sensor, the 8 working electrode, where we print this carbon electrode. Then we have the silver reference and contact, and then the dielectric here. And then we transfer it to the substrate. But again, these are, traditionally, these are all rigid, bulky, and planar device. So this is a rotating disc electrode. This is a glass pH electrode. So traditional electrochemistry is very rigid and bulky. Now our body is very soft and curved. If you come to visit us in San Diego Zoo, you see this flamingo are very stretchable. So biology is very soft and curved, stretchable skin and so on. So we need to to bridge the gap between the rigidity and softness of biology, developing devices which are flexible and stretchable. So we are spending a lot of time modifying our ink and the structure to make this stretchable serpentine structure. The ink is also modified to have ink which are stress enduring, so we can really stretch 200-300%. We can twist the device and push it to the limit. So here we pinch it, or in this video you can see we stretch and stretch 200-300%, keeping the conductivity, keeping the performance, twist it to the limit, and then we keep the performance under extreme strains. If you stretch too much, you have a crack. So we're also building self-healing capability in our ink. So as soon as you have a crack, it restores in 5 seconds. Here we have magnetic particle in the ink. So as soon as you have a crack, they're combined back magnetically to restore the performance. So having this capability, now we can go to actual sensing on the skin, for example. So we want to build a lab on the skin, both to do sweat monitoring or ISF monitoring, where we extract the ISF to the surface non-invasively. So I have completely non-invasive epidermal monitoring. So we go to the beach in San Diego, we get buying some tattoo paper, and then we print the sensor on a tattoo, which is transferred to the skin, like other tattoos. So we have nice looking flower electrode. These are the referent, working and counter electrode. These are the contact. We put the flexible electronic. And then we can twist it, as you can see here. So depending on the way you put it, you can monitor inside the sweat or outside to monitor the surrounding environment for security application. You can place it either way. And then we started 10 years ago working on lactate for performance monitoring, where lactate is a measure of the muscle fatigue and fitness. So we mobilize the lactate oxidase. This is my nano-engineering department. So these are the 3 electrodes working reference counter. We put the lactate oxidase here, and you start to exercise, you're getting the sweat. After 10 minutes, you see the profile of lactate. If you don't have the enzyme, you see the control, there is no interference. So this is typical amperometric calibration plot. This is in vitro, nice linear dependence, 1 to 20 millimolar, covering the sweat lactate range. More recently, we're starting now to combine the chemical marker with vital sign. So you can have on the same tattoo print the ECG sensor. If you see here, these are the ECG electrode, and these are the biosensor for glucose, lactate, glutamate, etc. So on the same device now we can monitor vital like ECG and the chemical marker like lactate or glucose without any crosstalk. There is no crosstalk between neighboring sensor, and the same wearable electronic to power both. So this is the first example of a hybrid patch, do the physical vital and the chemical marker. We're doing much more now. We do blood pressure with glucose. We do other vital with other biomarker. So as I mentioned, glucose is the biggest commercial potential monitoring, managing of diabetics. So you can do it in ISF, interstitial fluid, which is correlated nicely with the blood as well as the sweat. And again, we try to do it non-invasively, having our tattoo like this connected wirelessly to mobile device. So this is a tattoo paper that can do both the iontophoretic extraction in 90 seconds, then you do the detection in total in 2.5 minutes, non-invasive. You extract the ISF, bring the glucose to this electrode, and then you measure it amperometrically in less than 200 seconds. So the way you place it on the skin, it looks like this. This is the electronic, is reusable. The tattoo is single use for all day. Again, you can do the reverse iontophoretic here, and the detection. This is from the Technische Museum in Vienna, Austria. They put the diabetic plaster here and show it as the future technology in Vienna. But overall, using this, you can see, we don't have the video, but anyway, this shows that you place it very easily, and again, the electronic is reusable. Also, we can do the same for alcohol. So we have the nice dinner, we ate and drink. Now we can measure our alcohol. In this case, we stimulate the sweat. This is like in cystic fibrosis. You use pilocarpine to stimulate the sweat. The same type of tattoo, instead of extracting the ISF, you stimulate sweat. Sweat alcohol is correlated to blood alcohol. You have the enzyme, alcohol oxidase. You monitor the peroxide. And again, wirelessly tell you how drunk you are. So you see in 2 minutes, 1 minute extraction, 1 minute detection, tell you drunk, not drunk, drive, not drive. And again, this is typical response control, not drinking. And then when you drink, you have different level of BAC, and the current signal increase. So again, non-invasive. So again, you eat and drink, and you can measure both of them. This tattoo, this panda-shaped tattoo, can measure both your glucose and alcohol, and it can measure in two different biofluids. So you can measure the sweat alcohol on this electrode and the ISF glucose. So at the same time, when you apply the iontophoretic current, you have stimulated sweat here for the alcohol, and then the ISF glucose goes here. In 5 minutes now, you can measure both your alcohol and glucose. These are controlled, and no eating, no drinking, but when you eat and drink, you get a nice, and this is nicely correlated to the blood glucose meter or to traditional alcohol monitoring. So this example of monitoring the same tattoo, two different biofluid, non-invasive, the ISF extraction and the sweat stimulation. So we call it glucol. For glucose and alcohol, you can, following the dinner, you can measure both levels. Now you can go to my KGB device, this Potassium-Glucose-Biosensor on my glasses. So you put the potassium, K, on this sensor, this the electronic for potentiometric, on this Dolce Gabbana glasses. And here we have the glucose one, or lactate. So this is potentiometric for electrolyte. This is amperometric for metabolite, lactate, glucose. And again, you can easily replace this different sensor and get the response for potassium or glucose on this glasses device. So these are replaceable. You place it easily for multi-analyte detection. So we mentioned electrolyte. Again, pH of skin is the major interest. And again, this is the traditional decade-old, or a century-old glass pH electrode, very rigid, very big. We need to convert it to the skin using solid contact, printable ion-selective electrode. This is based on polyaniline, recognition of the proton. So we print this smiley pH sensor. These are the two electrodes, the reference, and working with the polyaniline recognition. We transfer it to the skin, still smiling nicely. You see the two electrodes. And then we can apply 50 repeated bending to the limit. After all this bending, it's still smiling and still responding. So this is the response before and after 50 repeated bending, strain. You keep the performance. The same is for sodium, for hydration monitoring. Again, 100 repeated stretching. This is a sodium textile sensor. Every 20 stretching or 20 bending, you see nice sodium performance, no carryover, a nice, what we call Nerstian performance, typical slope of around 60 millivolt per decade. We also pumping the sweat, so the sweat glands can push the sweat in this microfluidic device. So this is my epidermal microfluidic to allow to sweat to flow over the detector and remove and replenish the sweat to avoid any mixing on the sweat. So this is a PDMS flexible microfluidic with the electronic reusable, where 4 inlet will pushing the sweat over the detector and remove it from here. This is how we make it on PDMS. We place the sensor with the glucose oxidase or lactate oxidase. We can stretch it and twist it. And again, we do the exercise. Sweat is coming. We can measure the lactate again on the lactate oxidase sensor. These are two different subjects. You can do glucose, the same. Electrolyte. Here we have dye. The sweat is pushing the dye over the potassium and sodium sensor. So if sodium sensor and potassium sensor for sweat electrolyte, the sweat is flowing and pushing the dye for demonstration. So this was on the skin. Now we, again, the DOD is interested a lot of textile, also the sport, Nike and Adidas. They want to do a wellness and performance monitoring. So we have sensor again, lactate for performance, injury, nerve agent detection, all on textile. So again, we have to embed the sensor on different textile from socks to sleeve, t-shirt. We started 10 years ago with underwear on the elastic vest, good contact with the skin. This is sweat uric acid, what we call voltammetric detection on a printed carbon electrode. Again, we have to twist it. We have the serpentine structures that can stretch, so we stretch it, twist it, and they endure these repeated deformations. Again, we put the electrolyte sensor on different wristband, again on the underwear, so monitor sodium and potassium. We put this elastomer like Ecoflex or polyurethane to allow the stretching. So we have a joint reference electrode, potassium and sodium sensor, and you can see stretching 100, 200%. This serpentine structure is stretched. Also, the material is stretched by the polyurethane, which is elastomer, so you can do it on different textile like here. Bandage for wound healing. There's a lot of interest to managing the wound, monitor the wound, the healing process. pH is one parameter. Uric acid is another parameter. So this is our pH sensor on the bandage. With the polyaniline testing, you see 100 repeated bending of the bandage. This is the pH response. Nice carryover repeatability of the sensor. So all on a bandage with a reference and working ion selective electrode. Uric acid is another measure of the wound severity and the wound fluid. So we have the enzyme uricase immobilized here, and again monitor the wound healing progress, wireless transmitting this level of the uric acid in the wound fluid. And more recently we have this bandage for screening for melanoma. So in a melanoma mole, you'll have the enzyme tyrosinase is expressed in stage 2, stage 3 melanoma. So we immobilize the substrate catechol. In the presence of tyrosinase or the melanoma, it will convert the substrate to the benzoquinone, and we measure the benzoquinone reduction. It's all take less than 90 seconds. So when you go to dermatologist for your annual screening, you can quickly screen for potential presence of tyrosinase and melanoma in less than 2 minutes, while you're taking it for biopsy and take few days to validate it. But this is for quick screening. Again, reusable electronic single-use tattoo where you immobilize the substrate to look for enzymatic activity, a different stage of melanoma. We do a lot of glove-based sensing for the defense, starting with explosive residue, nerve agent residue, drug residue. So again, you swipe the surface for powder, let's say, of opioid or a pesticide or other nerve agent. On one finger, this is the collection finger, and then you close the two fingers, you have your electronic on a ring and monitor, let's say, the nerve agent that you collected. So the variety of paper on explosive, nerve agent, drug, gunshot residue. Overall, this will be a glove with, we have 2 gloves, 10 fingers, so we can have at least 8 to 9 sensors, one for collection and other 9 fingers for different analytes summarized in this review. So drug, nerve agent, explosive, pathogen, spices, as you'll see later on. So it's chemistry at the tip and touch. This is the example of the nerve agent. You see we have the serpentine structure to allow you to stretch the structure. So one finger for collecting the pesticide or the nerve agent. The second finger with the enzyme organophosphorus hydrolase. So you bring the collected powder, you close, you develop the product of the nerve agent, you detect the nitrophenol product or the fluoride product of sarin and somen. Opioid we are doing recently, fentanyl detection using this sampling glove. You get nice response at the nanomolar level of fentanyl. And this work with Honda in Japan, they want this Robotic to test different flavor, spiciness, sweetness, sour, Again, different finger to measure the capsicum level of spiciness, tell you if your coffee is decaffeinated or if you have sugar, and again, this is the capsicum chili. Another security device is this ring, this nice, not diamond, it's zirconia, but we have sensor for explosive, nitroaromatic vapor or peroxide-based improvised explosive on this side, and this is the nerve agent, so threat detection on the ring. All the electronic is placed inside. Oh, I don't have the slide, but the electronic is inside, and you can monitor both the explosive vapor and the nerve agent vapor. So microneedle allow you to go under the skin to monitor the ISF, which ISF have a very good correlation to blood, especially for small molecule. And we can build an array of microneedle. Here we have 12 microneedle, which are individually addressable, can have lactate, glucose, sodium, glutamate, etc. All on this is only 8 millimeters square. These are 9 microneedle here. And again, are minimally invasive, pain-free monitoring. The way we assemble it with the electronic on top, and this is the size. So we started the journey again a decade ago of lactate and glucose monitoring on this hollow microneedle. We're filling them with paste containing glucose oxidase or lactate oxidase. Nice linearity, nice selectivity. So a lot of interest in the CGM glucose monitoring. This is a self-powered glucose sensor with the microneedle acting as a biofuel cell. Glucose is the fuel, and then oxygen is on the cathode. So you get power which is proportional to the glucose fuel concentration, which can act as self-powered glucose sensor without the need for a battery, because the power is proportional to the glucose fuel concentration. More recently, we pushed the diabetic management to add another sensor for the keto, or the beta-hydroxybutyrate is important diabetic biomarker. In addition to glucose, on the same patch, you have the glucose oxidase sensor and the keto sensor neighboring, and monitor simultaneously the ketoacidosis, which is extreme complication of diabetic, along with the glucose. And now we are adding more electrodes like alcohol and lactate to have more comprehensive monitoring and management of diabetic, even adding cortisol and insulin to extend it for optimal insulin delivery. This is the alcohol sensor. Again, we have hollow microneedle, platinum. On top we have alcohol oxidase, we have perm-selective layer. This is penetrating the skin. L-DOPA monitoring for Parkinson management, extremely important to monitor the level because of the neurotherapeutic range of L-DOPA. So on the same microneedle, we have orthogonal detection, one based on enzyme and one is non-enzymatic. This is the enzymatic, where the enzyme tyrosinase convert the L-DOPA to dopaquinone. We monitor the product, or you can do direct measurement, voltammetric. On the same microneedle, you do both of this, and we're aiming for human subject testing. So the goal is to combine this microneedle with drug delivery autonomously, where the sensor input will activate, like in a logic gate approach. Here we have an N gate, where you have 2 biomarkers, let's say for cardiac, you can have a lactate and lactate dehydrogenase. You get 1-1 for N gate, it will open the gate to release the drug. Only when you have 1-1, the 2 biomarkers will open and close. These are conducting polymer actuators that can open and close in response feedback of this enzyme logic sensing. So we're working a lot with DITRA for both organophosphorus nerve agent and also opioid detection with soldier, not only for sensing but also for drug delivery. This is combinatorial delivery on the same 2 millimeters square. We have many needles for different drug. One is fast delivery, one is sustained delivery, both for the nerve agent and also for the opioid. Here we have example when we open and close different drug reservoir. You will see here we have the right side is open, then both sides are open, then we close the right and open the left one. This is using dyes. We illustrate this delivery of multiple drug on demand based on the sensor response, sense and act. Moving to saliva, the saliva is rich with a stress marker like cortisol, performance like lactate. So we introduced this mouse guard sensor, can do all saliva monitoring in the mouse. This is the early prototype 2014. Now we're sealing everything for safety. And the early work was for lactate monitoring, again for performance. With lactate oxidase, giving you the peroxide product, uric acid for gut disease, is our spin-off company, MouseSense in Carlsbad. So the signal is wireless transmitted. This is the uric acid product of the uric acid reaction. You get the peroxide all transmitted to a mobile device. And so we encapsulate everything for a variety of defense monitoring. This part of the NextFlex DoD activity, the Air Force in Dayton evaluating for pilot performance. Another way to do the saliva for newborn baby, we put our sensor on a pacifier. So the hypoglycemia is a major problem with newborn tiny babies, and instead of wire them or put mouse guard, we give them the pacifier, and there is fluidic, and the sensor is outside of the mouse. So just snipping of the pacifier, pushing the saliva one way to the sensor, and we evaluated it for diabetic before and after the meal. This just came out in analytical chemistry, well correlated to blood glucose. Well, the baby, they need to monitor an hypoglycemia every hour. Normally they're taking the blood, which is very painful for this tiny newborn. So all of these devices need energy, and we need to meet the energy needs of this wearable device. We're developing energy harvesting like biofuel cell or piezo, and also energy storage, supercapacitor and battery, and we combine the harvester and the storage in a printable manner. So again, we have using the sweat lactate is the fuel. These are biofuel cell. We have few millimolar of lactate or glucose in sweat, and then oxygen on the cathode. So there's plenty of sweat at the bottom in our socks. So we developed this again serpentine-like anode and cathode with lactate oxidase, and this can generate few milliwatts of sweat. Again, this is a device where we have a pellet of lactate oxidase and the serpentine bridges that can stretch. And this from sweat lactate you can get a lot of, I don't have the video for this, but basically it will show the stretching and powering of device, giving you up to 2-3 milliwatts. Then we combine it with the supercapacitor here to store this energy. So on the same textile, we have on one side we have the biofuel cell, the other side we have the supercapacitor for storing this energy. Again, highly stretchable, as you can see here, twistable. So you generate a lot of the power from the sweat lactate on this inner device, the outer one, store it, and you can have a complete energy power system with harvester and storage are integrated. We can replace the supercapacitor with battery. These are zinc manganese battery or zinc silver oxide battery, all on textile, that we also use to combine with a piezo harvester or a biofuel cell. So either supercapacitor or battery for storage, and then different energy harvesting from sweat, from light, or from motion. So we're addressing the energy issue, the data security. So all of this capability give us a real-time continuous monitor of chemical marker, both on our skin, in the surrounding environment, underwater. We work with the Navy on wetsuit of the diver. So we monitor electrolyte, metabolite, Again, glucose is a major initiative, security application, lactate, smart, fitness, hydration, all for 24/7 continuous non-invasive chemical monitoring. So again, this is thanks to my large group of 50 people that did make it all possible. Very talented postdoc and student and a visitor, funding for various agency, company, defense, NIH. And thank you all for your attention. Thank you very much. Mustafa. Yeah, it's a different story. The nanomotor we can tell you next time. It's also exciting.
And I'm glad that you closed your presentation with the power issue. You know, you discussed biocells, supercapacitor. As we embed more of these sensors, either subcutaneously or the surface of the skin, for real-time measurement, power becomes critical, as well as communication from the sensor to whatever device is communicating to. Where do you see the future of resolving the power issue with these sophisticated and multimodality sensors that are being added?
But we do also multimodality energy, so we harvest energy from different sources, from motion, this tribo energy or bioenergy of sweat lactate, even light, and then coupled with different storage, supercapacitor, battery, all in the integrated multimodality addressing the energy. And even using it as self-power sensing. We use the power as a signal, so you get the signal which is proportional to glucose or lactate, so you eliminate the need for batteries.
Thank you.
In the back here.
Thank you for a great presentation. My focus is completely on the industry side and in dealing with elder populations. And, you know, one of the challenges when I look at the amazing research you guys are doing is, of course, all the postdoc students are young. So these tattoos are on skin that's not wrinkled and isn't really sensitive and isn't the tissue paper thin that you get in the elder population. Are you guys, or do you know anybody else who's doing any work with my favorite demographic?
Yeah, I mean, in Japan, the Japanese colleague of mine, the elderly population in Japan is growing rapidly. Again, looking for fall prediction. So this electrolyte imbalance, sodium, potassium, can predict the fall. Again, a lot of the elderly have chronic disease like diabetics, a lot of this. So this is remote monitoring also in the US, lowering the cost. So in remote location, you can go to the nurse all the data. So there's a lot of interest indeed for the elderly. So as I mentioned, start from the newborn, from the baby, all the way to the elderly. Yeah. Yeah, please.
Sorry, one more, kind of along those lines. Do you know anybody who's doing work on CO2 sensing in the blood? Especially, you know, like, again, going back to the elderly population and a COPD prevalence of disease, that's one of those markers that says where you are in COPD progression.
Yeah, so CO2 is more challenging. I mean, again, a lot of pH, hydration, electrolyte, metabolite, but CO2 will be more challenging for continuous real time. ELISA, many of these protein biomarker, you need all the washing and tagging, but these days our new aptamer can allow you to do protein. You can even working on insulin for diabetic. Insulin become important parameter for managing diabetic to monitor the level which are in the picomolar. So, but you need an ELISA, it's very challenging. But there are new aptamer, switchable aptamer, can do real-time cortisol and insulin. We are working on this.
A very good presentation. I was just wondering about, large-scale manufacturing. So, in some of your use cases, you've shown traditional PCBs going alongside your sensors. What do you foresee in terms of the scalability of flexible electronics, where it is currently, and in terms of, scaling so that you can now improve the form factor along with your electrochemical sensing?
No, that's why we picked the printable. The screen printing is large-scale production at low cost, and we impart this mechanical stretchability, but it's the same printing that you use to make the glucose strip, billion of these every year, so we can readily, there is automated screen printing that you can do it at large
do
scale.
they have the same power of computing, computational power and other resources that are currently available with, let's say, traditional silicon chips? And
so even the electronic control, I mean, the electronic is reusable. The sensors are disposable, so you need less of this. But also the electronic, we have the battery is included, and we have all this self power capability and make it flexible, mechanically deformed. So yeah, all of that will be included. Data treatment, data security is a big concern for remote monitoring or wireless transmission of data, let's say the elderly, or even to foul play for glucose. If it's closed loop, you don't want to sabotage the insulin delivery, etc. So data security become a major concern as well.
I have a somewhat similar question. I'm involved in the commercialization of academic technology into the commercial consumer market. I look at all these wonderful technologies and I think, wow. Then I look at the market and I say, where are they, right? I saw some of the slides that were 2010 with printable things on the underwear band, and I say, boy, that seems like such a great idea. What are some of the barriers to having some of these technologies commercialized, and why aren't we seeing more of them on the market?
No, it's a good point. And again, we have a 4 spin-off company. What you show here, we have spin-off company for the mouse guard. We have a microneedle for glucose. BioLink is a method to do clinical trial. For the fluidic, we have a company in Sweden. So 4 of these technology are already spin-off. The barrier, as I mentioned, large-scale validation. If it's for FDA, it takes time, So even the glucose is a second phase of clinical trial. A lot of the sport, Adidas, Nike, is interesting in the textile-based sensor for performance monitoring. Yeah, but there are still the challenge of stability. If you do it for prolonged, all these enzymes are not robust. So but the best example is the CGM, the glucose. You have 4 companies like Dexcom and Abbott and Medtronic. This is already the CGM market is over $1 billion, and there are bigger needles. There are 5 millimeter, 10 millimeter. We are now moving to smaller needle, microneedle. But glucose is the nice example of the market. When you have a market like diabetic, it's pushing the technology, and you see, so company from Medtronic, Abbott have the Libra now. It's for 2 weeks. You can see the glucose sensor. Again, lactate, the market is not as big, so all the big players pushing the diabetic because of the market. But it's exciting. It's moving forward. Again, the sport company, Adidas, Nike, Under Armour, they're all interesting. The military, but the market is limited. For explosive or nerve agent, they're limited market. But the Air Force and Navy, they're all considering this. Yeah.
Hi, Tamir Dada, Feinstein Institute for Medical Research. So following up on sort of the last 2 questions, you showed many really cool electrochemical sensors, and then you showed the readout electronics. And about 7 years ago, I worked on some stretchable electronics. The main challenge that we had at that time was bridging this abrupt change in stretchability between your flexible tattoo and the readout electronics, right? So that's where the stress concentrates. How have you solved that problem, or do you not really see that as a problem in the applications that you envision?
No, I'm the director of a center on wearable, CWS, Center on Wearable Sensor. We have 20 faculty, including 8 electrical engineers. They are very good in integrating my device with a flexible electronic for wireless transmission, and direct integration is on textile or on tattoo or in the mouse. You saw the mouse guard is fully sealed with wireless electronics. So we are working closely, material scientists, electrical engineers, sensor people, data people. All of this is a multidisciplinary approach to build a system like this. Yeah.